What a wonderful continuation of our theme, making the invisible visible in the world of research, academic research. So following up Dr. Jennifer McIntyre, we're so fortunate to have Dr. Pat Tomco from our very own local area, University of Alaska Anchorage. Dr. Tomco is a professor of chemistry and director of the Applied Science, Engineering, and Technology Lab at UAA, and he's going to speak with us today about an update on the status of our knowledge about 6PPD-quinone in Alaska. So bringing it home. Please welcome Dr. Tomco.
Right. Good morning, everybody. Thank you so much for the invitation to come give a talk here. It was a great talk from Jen. When they're putting this conference together, they had asked Jen about giving a talk on 6PPD-Q, and immediately some of these questions went directly towards Alaska. So what type of research is happening in Alaska on this specific topic? What has been done? What is currently being done? And what are we hoping to accomplish here in the future? And so that's something that I've been very fortunate in giving the liberty to give you guys an assessment on. A lot of these are collaborative efforts with faculty all across the state of Alaska and different federal and state and university partners. But talking about the research is something I always have a chance to. I appreciate the chance to do. So just for those who may not know me, I grew up here in Palmer. I was actually in a band at Palmer High School, and we played a concert at this venue here in 2000. So, and then I've had friends who've gotten married here, I've been to funerals here. And so, being at a science conference here, this is a different world I feel like I'm in. So I guess it's one of my bucket list items, I guess, to give a talk. But now I find myself in Anchorage these days, even though I grew up in Palmer. I'm at UAA. I'm in the chemistry department. I'm a professor of chemistry. And so is a photo of our lab. We're at the ConocoPhillips Integrated Science Building. It's a pretty modern lab. A lot of people don't recognize just all the facilities that are present here locally that are available for conducting research. And I just want to spend a couple minutes kind of talking about that. But the research I'm going to talk to you about here is a collaboration with several other faculty. Dr. Zach Redman, Dr. Jonathan Stesick. Zach's in the chemistry department. Jonathan's in the biology department. Aaron Larson, who's here in the audience. We have Ben Barst at UAF. Brian Demento, who I don't think I got a chance to put his name on the slide here. And a lot of undergraduate students. So we work a lot hand in hand with undergraduate students, and it's just a pleasure to get a chance to work with all of them. And the lab that I am the manager of is the Applied Science, Engineering, and Technology Lab. It's an analytical chemistry facility. You guys might not realize that, the largest chemistry facility we have in the whole state is right over here at UAA. We have over $7 million worth of nice analytical equipment. Jen talked a lot about these very fancy high-resolution mass spec screening approaches so that you can detect and identify novel compounds in water, where you have like contaminants, and there are known contaminants that you need to identify and quantify so that you can comply with regulations. We do that work, but we also do a lot of screening and detection of these novel contaminants that are not on a compliance list that you may not know that you already have. So we specialize in organic spectroscopy, inorganic, which is metals. So a lot of the common work that you heard from Jen here already. So, like I mentioned, I want to kind of bring this conversation back to the United States of America here. We heard a lot of talk about, Pacific Northwest, but, when I think of the U.S., there's one really big map up here. And, I always had that map on the wall, as a kid, and it was always, the opposite direction. And you can kind of see there's, San Francisco in the corner with that little, Mississippi River water here. But, where we really want to focus on is the work that we've known about here in the state of Alaska. And in particularly, I've been asked to talk about this 6PPD-quinone and the research that we've been doing. And that can be kind of categorized to 5 different topics. My background is environmental chemist. I like to focus on the chemicals that are present in the environment. I'm interested in the attenuation and the persistence of the chemicals because in Alaska it's cold, it's dark in the wintertime. We have a lot of snow. There's snow dumps. People collect large amounts and they store it in areas. And when the temperatures get cold and when the light gets dark, those are the perfect conditions for chemicals to be persistent. And these are oftentimes acknowledged in many of the federal requirements, but they are not actually tested. And so what a lot of the work that I have experience in is looking at how these chemicals behave in high latitude, subarctic, and Arctic conditions, and to see how, and oftentimes you see very different behavior. And what's really interesting to me is that sometimes contaminants are more persistent in high latitudes, but also really interesting to me is that sometimes they are less persistent. They degrade faster. So I'll give a slide about 6-PPD-quinone related to that. We've also done some work with Alaskan salmon acute toxicity. With coho, Chinook, and sockeye. I'll get a chance to talk to you guys about that. And then we've been doing a lot of work with the sublethal work, where we start with the premise that temperatures of streams are rising. Salmon are particularly susceptible to raises in temperatures. And so what is the co-exposure of increased temperatures and 6-PPD-quinone at these sublethal levels? What impacts might that have towards salmon? And of course, as was very eloquently pointed out, how species specific are these impacts towards sockeye, towards coho, and toward king as well. And then prevalence, I'll also give you a little primer about. We're interested in 6-PPD-Q monitoring, but the source of 6-PPD-Q is ultimately in these tire wear particles, and so we're interested in detecting and quantifying the reservoirs of these tire wear particles, specifically within Alaska. So is 6-PPD-Q in Alaska? The answer is definitively yes, it is. This is a slide from Birgit Hagedorn with the Anchorage Waterways Council. This was taken in 2021, a couple years ago. These are taken from 2 sites. One's at the Seward Highway at Chester Creek, and that the other one is this one is near C Street Bridge, where the storm drain comes straight off of the highway. And then also at Ship Creek at another storm drain, right where it comes straight off the highway. And this slide was shown in the last presentation here, but the LC50, or the concentration that kills 50% of the coho salmon, 95 parts per trillion levels. And so I put that in a big red bar on the levels here, and you can see the levels are significantly above that. And there were 2 sampling events, one in April and one in July. And when you look at the Seward Highway site, and these were kind of opportunistically harvested right ahead of a known storm event, and these were taken straight out of the storm drain. So when they get into the river, they will dilute further. So that's a caveat to this. But when you sample at stormwater, it's we've got up to 1,200 parts per trillion in the water during just following a storm event. Lower in the summer times, but still above that LC50 threshold level. And then when you look at these criteria, for 100, for even 10 parts per trillion, that's something that we should really should be aware of. So we asked ourselves, given that it's here, given that it's present, the fish hatchery was really interested to know about this. So we talked with David Starzinski, who manages the William Jack Hernandez fish hatchery out in Ship Creek, and we said, hey, we'd like to do some toxicity studies involving salmon. They have some Little Sioux brood stock from coho as well as Chinook. They don't have sockeye, but they also have, you guys probably know better than I do, but they have rainbow trout, Arctic char, and sockeye. We were able to get that from the Galena fish hatchery. So our idea was an aquarium-based toxicity test. We wanted to assess first, are the populations of salmon in Alaska more susceptible or less susceptible to 6-PPD-quinone? And we had, it was right at the end of the finals week, which I'm an academic. I operate on an academic calendar. So right around in May was when all the salmon became available, and they're happy to donate as much of each of these as we needed. We took them in buckets over to the vivarium at UAA and conducted these 24-hour static exposures. One thing that I thought that was really interesting to me was that dosing the 6-PPD-Q was actually quite difficult because it kept sticking to the containers of the walls that we were trying to analyze. And so our first step with this was trying to keep it in solution. I thought it was kind of ironic because you're finding it dissolved and you're finding it in the waterway, but we had such troubles of getting it to stick to the walls of the containers that we had to get the right aquariums to in order to get it to in order to get the chemical into solution just for these tox tests. And so we did these, we did this preliminary study at, 1 hour post spike and 24 hours post spike, and then we extracted the residues from the water, and then we quantify this on our mass spec, and we compared these two. And we found that, and this is the concentration, the concentration of the 6-PPD-Q that was added to the aquarium, and then the percent recovery. So what you're hoping for is kind of what that blue bar is showing is 100%. You're showing that if you're adding it to the aquarium, you're hoping to recover 100% of it back. We found that after about an hour, you can still recover it. But if you let it sit in your aquarium for over a day, you can get about 50 to 60% recovery of this. And so note that many of these tox studies, they report the nominal concentration or the amount that was added to these, but the concentration needs to be monitored throughout the course of this study. So in other case, we're able to correct for this. But the numbers that we present, these are the nominal concentrations, the 1 hour post spike. That's what we're reporting on, and so it's actually a conservative estimate of it. So this work was conducted with Dr. Stesic and myself. This is an acute lethality test. We've got Fry from Ship Creek as well as Sockeye, like I mentioned, from Galena. They're two very different sizes. One was, the 3 grams were the coho. They're about yay big. And then the little, 400 milligram, little tiny sockeye salmon. They could still open them up and they could still pull out all the livers and gill tissues. They were, the students were amazing in being able to do that. Duration of 24 hours, 24-hour exposure at 5 Celsius. And then the aquarium water, it took us a lot to dial the chemistry of the aquarium water that allowed them to live in these fish tanks. And our endpoint that we were studying was mortality. So what we did see is we did see mortality in coho. Our LC50 value was 67 parts per trillion. And so note that when you see 95 parts per trillion, we found that the coho salmon in Alaska are more sensitive to the 6-PPD-Q than was reported in the other studies. There are several studies, some have reported it as sensitive as this, but the take-home point is that these are on the more sensitive end of the spectrum, the Alaskan particular coho. Sockeye and Chinook, as was consistent with other studies, we wanted to reveal this and we wanted to assess this, and we were happy to confirm that both Sockeye and Chinook were highly tolerant to 6-PPD-Q. We didn't have any mortality, and we said our LC50 were above over 117,000 parts per trillion. That was actually the allowable level that our IOCOOK protocols allowed us to test up into. You could actually see the water change colors, and the fish were, the sockeye were doing just fine. So like I mentioned, some of these tests that we were interested in doing is this CT max, or the critical temperature maximum. And what we're doing is we're taking the temperature in these aquarium and we're ramping the temperature up slowly. And eventually, eventually the fish will reach kind of this thermal equilibrium where they do reach a point, there's a physiological death, there's a temperature at which they're going to die, but there's this endpoint or a loss of equilibrium. And so after their acclimation temperature, there's a temperature at which you can rise it to where they will kind of lose their equilibrium and flip upside down. When you take the temperature back, they come right back. You're not killing the fish, but you can actually, identify what temperature at which they start to lose their thermal equilibrium or their ability to tolerate thermal stress. And so we ramp this up to 30 C at, a slow ramp rate. And how long they were upside down for 5 seconds. And that's what we use to calculate this. And so again, we did this at 3 levels. One was the control, which received only water. One was ethanol, because in order to get the 6-PPD-Q dissolved, you had to put it into a solvent that you would add into the aquarium. And then we did this at the LD10 level. So the concentration that we knew would kill 10%. And that's one where we know that they're being stressed out, but it's not enough to kill the majority of the population. So when you look at the different species, you have coho, sockeye, and Chinook. The coho was interesting. We found that there was up to 1 to 2 degrees Celsius lower temperature at which the coho would lose their thermal equilibrium and flip upside down. And this is when exposed to that 39.9 parts per trillion level. At these very low levels, 15 parts per trillion, and in the ethanol solvent blank, you didn't, those were all consistent with the controls. Interestingly, the sockeye had the opposite effect. They actually increased their thermal tolerance following exposure. We call this a hormetic response. So whenever you're exposed to some, mild level of stressor, we think the idea is that these fish are essentially priming their system. They're essentially upregulating and a lot of metabolic function that's able to deal with a stressor. And so we think that they're highly able to tolerate it that way. Chinook, we didn't find any change on that. We've done some respiratory performance work where you get to take all the fish and you take the fish after the 24-hour exposure, you move them over into a bucket and you go chase them around. You pretend you're a predator and you kind of put your hand in the bucket and gonna go chase them all around and you exhaust them. And after they're exhausted, then you take the fish and you move them to another chamber. And then for 24 hours, you have this chamber that's monitoring the dissolved oxygen levels. And that's going to tell you about the oxygen consumption rate. And so we had thought that the exposure to this 6-PPD-Q would cause a decrease in the aerobic scope. So just the ability to handle these these aerobic activities, which are essential towards evading predators or swimming up a stream or any of these other behavioral endpoints. And we wanted to conduct this again at this sublethal level, asking ourselves, if you're exposed to this at this concentration, is that going to have any impact? This is kind of what the curves look like, where you have your control, your ethanol, and your 6-PPD-Q. After they are all swam out and you move them over, you look at the oxygen consumption rate, and that's going to be the absolute highest. And then you see how that oxygen consumption rate drops, and then you wait until it's in its 10th percentile level, and then that's where you take the mean of all of those measurements. You can see on a 24-hour time scale, you can kind of see at the very end, it kind of goes, whoop, it goes up a little bit. Why would that go up a little bit? Well, these are 24-hour studies. And so you start to see people come into the lab the next day and they start turning the lights on, they start closing the doors, and then, they all get a little stressed out. And so we don't take that time point. We use it at an earlier time point than that. But again, if we look at the respiratory performance, we find that there's the maximum, which was that first time point that I showed you. Those are affected in the coho salmon, is that oxygen consumption rate is lower, and in the resting rates, those ones are similar to the control level. And so the aerobic scope, in other words, the range at which you can tolerate this aerobic activity. I drove out here in a CRV, that thing goes up to, maybe it goes to 80 miles an hour or something, it doesn't go very far. It doesn't have a very large scope. It goes from 0 to 80, but if I've got a big Ferrari, I can go all the way up to, 300, probably not 300, but I can go a lot faster in a Ferrari. So, the larger the aerobic scope is, just the more aerobic activity and the capacity the fish has to conduct all these activities, like avoid predators or swim up a stream. And we found that was compromised in those which were exposed to these LD10 levels of the 6-PPD-Q. Sockeye seemed to tolerate it just fine. We didn't find any difference in the max or the rest or the aerobic scope. So the sockeye seemed to be very tolerant of it. One slide. This is an experiment I was told not to talk about, but I decided to talk about it anyways. The mitochondrial function. So the electron transport chain inhibition. There had been some studies recently that indicated that exposure to the 6-PPD-Q had some inhibition. This was using some rainbow trout, gill, and liver tissues. And so we wanted to start to test this on some of these exposed tissues that we had. And we looked at all the different complexes in the electron transport chain that are, to see if this mitochondrial inhibition was present in with the 6-PPD-Q. We did this with liver tissues, muscle, and gill tissue. To conduct this, you do it's called a Western blot. Western blot calculates the concentration of all the proteins, and it's all normalized. You essentially look at the abundance of each of the proteins in each one of these complexes. I was told not to give a talk. I was told not to talk about this because it was insignificant. There was no significance, which again doesn't paint the whole story. Each one of these graphs indicates liver, muscle, and gill tissue. And if you look at the differences between the control, the ethanol exposed, and the LD10 values, there's no differences at each one of those. So you can see the protein levels change between each of those complexes, but there's no differences between the 6-PPD-Q exposure. Now that just means that so there's the caveat, the protein levels are not different between the two, but it does not have any indication of the function of that protein. And so that's a study that we're currently working on, is whether these mitochondrial functions are actually inhibited. This was just simply an abundance of those proteins. Okay. I get to talk about something a little bit more in my wheelhouse, which is environmental persistence and sunlight degradation. Alaska is very cold in the winter, and it's also very dark. And like I mentioned, those are conditions to allow contaminants to be very persistent. We are interested in how persistent 6-PPD-Q is once it is liberated and released into the environment, and specifically when sunlight impacts it. Sunlight is a known way that chemicals can break down in the environment, and they can do it through two ways. Either the chemical itself can absorb sunlight, or the sunlight can be, or something else in the water. So organic matter or some other species in the water can absorb sunlight, and that can turn into a reactive species. And it can create, either of those can create what's called reactive oxygen species. And those reactive oxygen species then break the chemical down. Because if it's not persistent, it's not as much, it's still a concern, but there are less of a concern if it's easily broken down. And then this other question of transformation products, this 6-PPD-Q is a transformation product itself. Once it's broken down, is it turning into other transformation products? Just in case you missed that from Jen's slide, this chemical's red, and it's actually this is what it looks like when you bring up a solution of it in methanol. It's red. It absorbs light in the visible region. And this is the map of light absorption at different wavelengths. And so you can see the absorption of the light of the 6-PPD-Q. And then this other map that kind of has all these little zigzags on it, that's the light of sunlight. That's the light absorption that sunlight has. And what you see is those two cross over. So where the chemical is absorbing light and where the sunlight exists, where those two intersect with each other, that means that there could be a potential degradation of that compound. This is a very technical slide, and I'm going to drag you through it, and I apologize in advance for it. We have all these different probe molecules that can isolate different features and react with these reactive oxygen species and pull them out of solution. So we can start to identify what are the chemicals that are present in the water that are most responsible for causing the 6-PPD-Q to break down. What we're seeing on the y-axis is the rate. So larger bar means that the chemical breaks down faster. And what we're seeing are different treatments. So this one on the left here is called SRFA. That's organic matter. It's kind of the synthetic organic matter that we add to it. And we do this at 3 different temperatures. So 20 Celsius, 12, and 4. And what we're seeing here is kind of an interesting trend. The colder temperatures make the chemical degrade faster than in the higher temperatures. You would think if the temperatures go up, you would think that the chemical would degrade degrade faster, but actually it's the opposite. We find that at lower temperatures, that's actually causing it to break down. However, when you start to pull out the species that are causing it to break down in solution, you can get it to break down consistently. So you add IPA or isopropyl alcohol. What is causing this to break down? It's the reactive oxygen species in the water. This hydroxyl radical. So when you add something that removes the hydroxyl radical, you find that rate drops significantly. So on the right side, we look at the water, and then we had a bunch of water samples that we got from Fish and Game. We were doing some rotenone screening for them. These samples didn't have any rotenone in them, but we screened this lake water where we added the 6-PPD-Q, and we asked, is the breakdown rate going to be different in lake water compared to pure water? And we found that it wasn't. And what that's telling us is that these hydroxyl radicals essentially are being scavenged. And so the chemistry of the lake water, the chemistry of the stream water has a very big impact on whether the compound is going to degrade or not. So up at that take-home point that I have in the corner, it decays actually quite slowly. 4% per hour is the rate at which we found it to decay. And that's under its some blaring, strong lights. It's not in like a turbid environment or shaded or anything like that. We found that, yeah, it's a very long decay rate. So it's very persistent here, but under the appropriate conditions, you know, where you have the right water chemistry and the temperatures are lower, you can actually get the chemical to break down faster. Some other work we've been doing is the prevalence of these tire wear particles. Like I said, we see these as a reservoir of the 6-ppdq, and wherever you find a tire wear particle, you can also find 6-ppdq present. And so we're interested in identifying, is there rubber on the road? Is there, you know, can you quantify the mass of the tire wear particle as you scrape it off the side of the road? And to do this, we do some fun GCMS work, but we have to ask ourselves, what are the composition of these polymers in the tire wear formulation? And there are 3 essential polymers that are found, and everybody's got their own proprietary mix of it, all the different vendors, and they're all very secretive about like what those particular mixes are. But when you look at the polymers, it's essentially 3 different types. There's natural rubber, there's this synthetic material called butadiene rubber, and there's this third one here that's called styrene butadiene rubber. Now, when you put them into a GCMS, they pyrolyze, or in other words, they transform into these marker compounds, and they do this diagnostically. So natural rubber will actually turn into dipentene. Butadiene rubber will turn into this other chemical called 4-vinyl cyclohexene, and this styrene butadiene rubber will turn into these other marker compounds. And those are the ones, you can see the figure over here, you can actually monitor the abundance of those markers, and that quantity is actually going to tell you how much tire rubber is in your sample. So we've been able to, we went to Alaska Tire Service and just asked them. They were throwing away a lot of tires. They said, let us take all of your tires and analyze them. They said, I have no idea what you're doing, but okay. So they gave us some box cutters, and we were able to take some chunks out of it and then identify that, 2 of the polymer markers that we see in the literature are good markers for those, particularly in Alaska, but there's one of them, this 4-PCH, is not. And so this is specific to the formulation of the tire particles that are in Alaska. We see which ones are present, and now we feel comfortable with the database. Where we're applying this now are to these snow dumps, and I call snow dump ice core, kind of funny, but it's kind of forms a little ice core. But these, this is kind of what it looks like when we take the ice chunks out, and it persists all the way through May. Some ongoing studies that we're doing. We're also doing this work called metabolomics. So if you think of yourself as the combination of all the chemicals that are present within you, I could tell you a little bit about your health. We think about all the different chemicals that you have, all the different amino acids and all the proteins and the nucleic acids. And if you look at all of the sum of each one of those and you combine them all together, that can tell you a little bit about your metabolic health. That's what these studies essentially do, is they take these individuals and you profile all the metabolites that are there, and you look at the fingerprint of them, and you compare those fingerprints to those which are controlled or exposed at high levels. This is a study from another group, but we're applying this technique similarly to coho salmon, livers, and heart and gill and muscle tissues, which have all been exposed to the LD10 levels. And what that can tell you is specifically which metabolic pathways are altered. In this case, you can look at amino acid impairments in amino acid metabolism through specific compounds. And so that's an ongoing study we currently have going on. I was hoping to give some data on this one, just about there, but we hope to have some data out for that pretty soon. So just a real quick summary about what I think is really important: knowing the prevalence of the 6-PPD-Q, knowing the persistence of this, and then knowing the toxicity that is specifically relevant to Alaskan populations. So at that, I think I got my time is up here. I'd just say thank you guys so much for your attention, and I'll be happy to answer questions, but also be around here in the panel for other questions if those do come up. So thank you.
Thank you so much. Time for a couple questions. We're going to roll in.
On our earlier keynote speaker talked about the Seattle area and all of the urban complexes and all the pavement and all that kind of stuff. And you're bringing this whole discussion a little closer to home in Alaska, but you're still, it appears, concentrating on pavement-oriented highways. has anybody stopped to think whether there's any significant impact on all of our gravel roads in Alaska? We have a lot of gravel roads, and they wear tires just like pavement, but they wear them differently. And I'm just wondering if anybody's looked that direction yet.
Yeah, I started with an interest in this. So the initial funding for this project came from ConocoPhillips Arctic Science and Engineering Endowment. And I said, well, you're driving all these big trucks up in these pristine, sites off of gravel roads. And oftentimes you're driving your trucks through these rivers. That's something you should be concerned about. We have not, but to your question, we have not looked at these more porous surfaces such as gravel roads. We started with the asphalt, and that's an area that we definitely want to explore later. So we're moving there.
So I think it's fascinating that Tomco effectively put new stockers are going to show any effect. Is there any speculation on what's going on?
You saw, I saw that mitochondria slide where we keep looking in the literature and seeing what are these potential, what are these potential effects, and we start running these tests ourselves, and we don't have I don't have a clear answer for you. I mean, the mitochondrial function was one idea. Was one. The second one. The second one was this metabolic adaptation. So if you have evolved these enzymes that are capable of detoxifying evolutionarily, then you can upregulate the abundance of these enzymes to break it down quickly, just so that you can excrete it quicker. That's again just an idea of it. Coho do spend more time. You guys all know this better than I do, but like coho spend more time in the stream at the fry stage than the others, and so there's more time in the stream. There's quite a bit of time that's in the stream compared to others. So I don't know if that has anything to do with it. Again, just speculation at this point.
All right, round of applause. Thank you so much, Dr. Tomco.
Thanks.
Now this time we're ready to roll into our panel discussions. I'm very happy to invite our next moderator. All right, with our esteemed panel members, going to come on up, please, to the stage.